Hello and welcome to today's video on the exciting topic of drug discovery. In this video, we will explore the fascinating process of finding and developing new medications that can improve our health and save lives. So let's dive right in. Drug discovery is a complex and time-consuming process that involves extensive research and experimentation. It all begins with identifying a target, which is usually a specific protein or pathway in the body that is involved in a disease or condition. Scientists then search for molecules, often through thousands of compounds, that can interact with this target and potentially treat the disease. The drug discovery process is a complex, multi-stage journey that takes years to complete. The first crucial step is target identification and validation. This involves finding the specific biological targets, like proteins or enzymes, that are associated with the disease. Once targets are identified, scientists use various techniques like high-throughput screening to find potential drug candidates that interact with the target. These lead compounds then go through a process of optimization, where their properties like potency, selectivity, and pharmacokinetics are refined and improved. Extensive preclinical studies, both in vitro and in vivo, are then conducted to assess the safety, efficacy, and potential toxicity of the drug candidate. If the preclinical results are promising, the drug candidate then enters clinical trials, where its safety and efficacy are evaluated in human volunteers. Finally, if the clinical trials are successful, a comprehensive application is submitted to regulatory authorities for approval to bring the new drug to market. The goal of target identification is to find potential drug targets by studying the disease mechanisms and identifying the key molecules involved. Once a potential target is identified, the next step is target validation, confirming the target's role in the disease and ensuring that targeting it will have a therapeutic effect. Selectivity is critical. We need to ensure the drug specifically targets the intended molecule and avoids off-target effects, which is crucial for safety and efficacy. These three steps, target discovery, validation, and selectivity, are all essential in the drug development process. High-throughput screening is a critical step in identifying potential drug candidates. It allows us to rapidly test a large library of compounds against a specific target. Fragment-based drug discovery uses smaller molecules, called fragments, to identify starting points for drug development. This approach allows us to explore a wider chemical space. Virtual screening uses computational methods to predict the binding affinity of compounds to a target protein. This helps us prioritize and select compounds for experimental testing. Once a hit compound is identified, we undergo lead optimization, a process of modifying its structure to improve properties like potency, selectivity, and pharmacokinetics. A lead optimization is a crucial step in drug discovery, where we focus on enhancing the properties of promising drug candidates identified earlier. This involves improving the pharmacokinetic profile, how the drug is absorbed, distributed, metabolized, and excreted in the body. We also work on boosting the pharmacodynamic properties, making the drug more effective and potent. Importantly, we aim to minimize toxicity and side effects through safety optimization. Throughout this iterative process, we conduct structure-activity relationship studies to understand how the chemical structure of the compound relates to its biological activity. By synthesizing and testing various chemical analogs, we can identify modifications that improve the desired properties and reduce the undesirable ones. A preclinical development is a critical phase where we evaluate the safety and efficacy of a drug candidate in animals. This involves a series of tests to assess potential toxicity and determine the optimal dose range for clinical trials. We also analyze how the drug is absorbed, distributed, metabolized, and excreted in the body. This pharmacokinetic data is crucial for optimizing drug delivery and minimizing side effects. Pharmacodynamic studies help us understand how the drug interacts with the body's biological processes and mechanisms of action to determine its effectiveness and potential therapeutic benefits. Formulation development is key. We need to create an optimal drug formulation for administration, considering factors like stability, bioavailability, and ease of use. Finally, we must establish a robust manufacturing process to scale up production and ensure consistent quality and purity of the drug candidate. Clinical trials are critical for evaluating the safety and effectiveness of new drugs. They involve testing the drug in humans, typically across multiple phases. Each phase has specific objectives that build on the previous phase. Phase 1 focuses on establishing the drug's safety and determining the appropriate dosage. Phase 2 examines the drug's efficacy and continues to assess safety at the determined dose. Phase 3 involves large-scale studies to further evaluate the drug's efficacy and safety. Phase 4 is post-marketing surveillance to monitor the drug's long-term effects. The results of each phase inform the design and execution of the next phase of trials. Drug approval is a stringent process that requires rigorous scientific evaluation and adherence to strict regulatory standards. Extensive testing, including clinical trials and preclinical studies, is critical to assess the safety, efficacy, and potential side effects of a drug. Regulatory agencies, such as the FDA, play a crucial role in ensuring the safety and efficacy of drugs by providing government oversight throughout the approval process. The approval process is designed to protect public health and ensure that only safe and effective drugs reach the market. Pharmacokinetics is the study of how the body absorbs, distributes, metabolizes, and eliminates drugs. Understanding this is crucial to know how the drug moves through the body and how long its effects last. Pharmacodynamics explores how the drug interacts with its target molecules in the body to produce a therapeutic response. It looks at the mechanisms of action and the relationship between drug concentration and its effects. Together, pharmacokinetics and pharmacodynamics provide a comprehensive understanding of how a drug behaves in the body and how it produces its desired outcomes. This slide provides a high-level overview of these two important concepts in drug development and clinical use. The image helps visualize the journey of a drug molecule entering the body. Medicinal chemistry is a critical discipline in the drug discovery process. It involves designing, synthesizing, and characterizing new chemical compounds with desired pharmacological properties. Medicinal chemists leverage their expertise in chemistry, biology, and medicine to develop and optimize drug candidates. They use various techniques like computer-aided design, combinatorial chemistry, and high-throughput screening. Medicinal chemists play a vital role in identifying, characterizing, and optimizing drug candidates for therapeutic use. Their work is essential for developing new drugs to treat diseases. Computational drug design is a powerful approach that uses computers to simulate and analyze drug candidates. It combines computational chemistry, molecular modeling, and AI to process vast amounts of data on drug targets and potential drugs. This allows drug designers to predict the properties of new drug candidates and identify the most promising ones for further development. Computational methods work hand in hand with experimental data to accelerate the drug discovery process. The goal is to leverage the power of computers to design better, more effective drug candidates more efficiently. High-throughput screening is a critical process in drug discovery, allowing researchers to quickly test large libraries of compounds against a specific target. Automation and advanced robotics enable the screening of thousands or even millions of compounds, vastly improving efficiency and speed compared to manual methods. Data analysis techniques are used to analyze the results, identify promising hits, and optimize the screening process. This technology has revolutionized the early stages of drug discovery, accelerating the identification of potential drug candidates. Fragment-based drug discovery, FBDD, starts with small, simple molecules called fragments. These fragments bind weakly to a target protein, providing a starting point for drug development. FBDD often uses structural information from the target protein to identify fragments that fit into the protein's binding site. Once a fragment is found, scientists can use various techniques to modify and grow it into a larger, more potent drug candidate. FBDD has advantages over traditional drug discovery methods, such as increased efficiency and a higher chance of finding novel drug candidates. Phenotypic drug discovery is an approach that focuses on identifying drugs based on their effects on a biological system, rather than on a specific target. This is often used when the molecular target is unknown or difficult to identify. This approach allows for a broader search and can lead to the discovery of novel drug targets and mechanisms of action. It's a complementary approach to target-based drug discovery, which focuses on specific molecular targets. The goal is to identify compounds that have the desired biological effect, even if the exact mechanism is not yet understood. This can open up new avenues for drug development and uncover unexpected therapeutic opportunities. Biotherapeutics are a class of drugs that utilize proteins, antibodies, and other biological molecules to target disease processes. These therapies are often engineered for enhanced efficacy and specificity, potentially offering more targeted treatments with fewer side effects. Biotherapeutics encompass a wide range of approaches, including monoclonal antibodies, gene therapy, and cell therapies, expanding the possibilities for treating disease. Protein-based therapies, engineered biomolecules, and emerging treatment modalities like these are all part of the growing field of biotherapeutics in drug discovery. Cancer is a complex disease driven by various genetic and molecular changes in cells. Oncology drug discovery aims to identify key pathways and proteins involved in cancer cell growth, proliferation, and survival. By targeting these critical processes, we can develop drugs to disrupt and potentially halt cancer progression. Precision medicine is revolutionizing oncology, enabling personalized treatments tailored to the genetic and molecular profile of a patient's tumor. This allows us to select the most effective therapies for each individual, improving outcomes and minimizing side effects. Immunotherapy is an exciting new field that harnesses the power of the immune system to fight cancer. These therapies work by stimulating the body's own immune cells to recognize and attack cancer cells. Rigorous clinical trials are essential to assess the safety, efficacy, and optimal dosage of new cancer therapies before they can be approved for widespread use. These trials help ensure that new treatments are both safe and effective for patients. Neuroscience drug discovery is a critical field focused on developing treatments for a wide range of brain disorders. Identifying and validating specific targets within the brain is crucial for effective drug development. Understanding neurotransmitter pathways and their role in brain function is essential for developing drugs that modulate these pathways. Clinical trials are essential for evaluating the safety and efficacy of new drug candidates for treating brain disorders. This slide provides an overview of the key focus areas in neuroscience drug discovery, including brain disorders, target identification, neurotransmission, and clinical trials. Infectious diseases pose a significant threat to global health, which is why drug discovery plays a crucial role in combating these threats. The emergence of drug-resistant pathogens is a growing concern, so drug discovery efforts focus on developing new antibiotics and antivirals to address this issue. Vaccines are essential for preventing infectious diseases, and research and development of new vaccines continue to be a priority. Identifying new targets for drug discovery is essential, and research focuses on understanding the mechanisms of pathogen infection and developing novel therapies. Rare diseases affect millions worldwide, causing significant suffering. Drug discovery efforts are vital to provide effective treatments and improve patient outcomes. Developing drugs for rare diseases presents unique challenges, including limited patient populations, complex biology, and high development costs. Researchers are working to develop innovative, targeted therapies tailored to the underlying mechanisms of each rare disease. These efforts aim to alleviate suffering, enhance quality of life, and extend lifespans for rare disease patients. Translational medicine is the process of taking research findings and applying them to clinical practice, with the goal of developing new and more effective therapies for patients. Clinical trials are a critical part of this process, as they allow researchers to evaluate the safety and efficacy of new drug candidates before they can be approved for use. A key focus of translational medicine is adopting a patient-centric approach, which means deeply understanding the needs of patients and tailoring treatments accordingly. This helps ensure that new therapies are truly meeting the needs of the people who will use them. By bridging the gap between research and clinical practice, translational medicine plays a vital role in bringing important new treatments to those who need them. Biomarkers are measurable indicators that can predict disease outcomes and drug effectiveness. They help monitor disease progression, enabling personalized treatment based on individual patient responses. Certain biomarkers serve as targets for drug development, guiding the design and development of new drugs with greater precision. Biomarkers allow for early disease detection, providing a window for more effective treatment interventions. This slide provides an overview of the key ways biomarkers are used in drug discovery and development. Pharmacogenomics is the study of how genetic variations influence how people respond to drugs. This information can be used to tailor drug treatments to individual patients, improving the effectiveness of the treatment and reducing side effects. Personalized medicine goes beyond just pharmacogenomics. It encompasses a range of genomic approaches to provide the right treatment to the right patient at the right time. The goal of personalized medicine is to enable more effective and safer therapies by taking into account a patient's unique genetic makeup and other individual factors. This approach promises to transform healthcare by moving away from a one-size-fits-all model towards more targeted, individualized treatments. Drug repurposing involves finding new uses for existing, approved drugs. This can accelerate the drug discovery process since safety and efficacy data are already available. Repurposing can be a more cost-effective approach compared to developing entirely new drugs from scratch. Repurposing can expand treatment options and address unmet medical needs by providing alternative therapies for existing diseases. The drug discovery process is incredibly complex, with a high rate of failure that results in significant costs and delays. Identifying and validating suitable drug targets is crucial, but can be very challenging, especially for complex diseases. Emerging drug resistance, particularly in infectious diseases and cancer, poses a major obstacle to effective treatments. Ensuring the safety and efficacy of drugs is essential, but assessing potential toxicity and side effects can be extremely difficult. The challenges we face in drug discovery are significant, but by understanding these obstacles, we can work to overcome them and develop more effective and safer treatments. Drug discovery is being revolutionized by emerging technologies like AI, advanced imaging, genome editing, and automation. AI is identifying promising drug candidates and optimizing development processes, accelerating the discovery pipeline. Advanced imaging is enabling scientists to visualize biological processes at a cellular level, providing critical insights for drug development. Genome editing technologies like CRISPR offer the potential for precision targeting of disease-causing genes, paving the way for more personalized treatments. Automation and robotics are streamlining drug discovery workflows, increasing efficiency and throughput to bring new therapies to patients faster. These cutting-edge technologies are transforming every stage of the drug discovery process, from target identification to clinical trials. Collaboration is about sharing knowledge and resources with others to combine expertise and accelerate drug discovery. Open innovation involves external partnerships, leveraging ideas and technologies from academia, startups, and other industries. This approach fosters creativity and promotes the development of novel drug candidates. Collaborating and embracing open innovation can help drive innovation in the drug discovery process. Patents are essential for protecting the intellectual property of drug discovery companies, safeguarding their investments, and promoting innovation in the pharmaceutical industry. The patent process involves a rigorous legal framework, ensuring that inventions meet specific criteria for novelty, inventiveness, and industrial applicability. Patents provide incentives for pharmaceutical companies to invest in research and development, contributing to advancements in medicine and healthcare. Drug discovery is a critical but expensive process, requiring significant investments from various sources. Funding comes from pharmaceutical companies, venture capitalists, government grants, and philanthropic organizations. The average cost of developing a new drug can range from $1 billion to $2.6 billion, depending on the complexity and regulatory requirements. On average, it takes 5 years to develop a new drug, but the success rate is only around 10%. There are currently around 150 companies engaged in drug discovery, all working to bring new treatments to market. The high costs and long timelines involved highlight the challenges and risks inherent in this industry. The goal of this slide is to highlight some of the key ethical considerations in drug discovery and development. Informed consent is critical. We must ensure clinical trial participants fully understand the risks and potential benefits before enrolling. Fair access to new medicines is essential. We need to address disparities and make treatments available to all who need them. Protecting patient privacy and confidentiality throughout the process is of utmost importance. Data integrity and transparency are also vital, maintaining accurate, reliable data and fostering open communication. These ethical principles help guide us in developing new drugs in a responsible, patient-centric way. Artificial intelligence is revolutionizing drug discovery by analyzing vast amounts of data, identifying promising drug targets, and designing new drug molecules more efficiently. Personalized medicine is becoming increasingly important as we can now tailor treatments to individual patients based on their unique genetic makeup and other factors. Biomarkers are playing a crucial role in identifying patients who are most likely to benefit from a particular treatment, as well as monitoring the effectiveness of that therapy. Digital therapeutics are software-based interventions that can help manage chronic diseases and improve patient outcomes, complementing traditional pharmaceutical treatments.